This video tutorial is all about biotechnology and some of the uses of microbes in biotechnological processes. Commercial drug production uses large stainless steel containers called fermenters, and I've included an image of a fermenter on this slide, in which the growing conditions can be controlled to ensure the best possible yield of the product. The conditions that must be controlled include temperature, so you don't want it too hot or the enzymes will denature, but equally you don't want it too cold or the growth will be limited. Nutrients are made available, so microorganisms can grow and synthesize the product. Sources of carbon, nitrogen, minerals, and vitamins specifically are needed. Oxygen is made available because you want the microorganisms to respire aerobically. You want to adjust the pH, so enzyme activity is optimum, and hence sort of growth and synthesis aren't affected by extremes of pH. And you also want to be able to adjust the concentration of the product. So if the product is allowed to build up too much, it may affect the synthesis process. So a fermenter must first be sterilized using superheated steam. It can then be filled with all the components required for growth and supplied with a starter culture of the microorganism to be used. The culture will be left to grow and synthesize the product. Some products are synthesized by the microorganism during normal metabolism when they are actively growing. These are called primary metabolites. Such products are continually released actively from the cells and can be extracted continuously from the fermentation broth. The broth is topped up with nutrients as these are used by the microorganisms. Some of the broth is removed regularly to extract the product and remove cells from the broth. Otherwise, the population will become too damp. So this is known as continuous culture and keeps the microorganism growing at a specific growth rate. Other products are produced only when the cells are replaced or placed under stress, such as high population density or limited nutrient availability. These are called secondary metabolites and are produced mostly during the stationary phase of growth. And we'll look more at this in another video tutorial. During the stationary phase of growth, the culture is set up with a limited quantity of nutrients and allowed to ferment for a specific time. After this time, the fermenter is emptied and the product can be extracted from the culture. And that's known as batch production. Asepsis is ensuring that sterile conditions are maintained. The nutrient medium would also support the growth of unwanted microorganisms, which would reproduce and reduce production, because the unwanted microorganisms would compete with the cultured microorganisms for nutrients and space, would reduce the yield of useful products, would spoil the product, and they might even produce kind of toxic chemicals or destroy the cultured microorganisms and their product. In processes where foods or medical chemicals are produced, all products must be discarded if contamination by unwanted organisms occurs. Flory and Chain devised the process to successfully mass-produce penicillin through fermentation by the fungus penicillium. Modern strains of the fungus have been selectively bred to be more productive than earlier strains. Penicillin is a secondary metabolite. It is only produced once the population has reached a certain size. Therefore, penicillin is manufactured by batch culture. The fermenter is run for 6 to 8 days. The culture is then filtered to remove the cells. The antibiotic is precipitated as crystals by the addition of potassium compounds, the antibiotic may be modified by the action of other microorganisms or by chemical means. The antibiotic is then mixed with the inert substances and prepared for the administration in tablet form, as a syrup or in any form suitable for injection. Citric acid is widely used as a flavour enhancer, a preservative in manufactured foods, and an antioxidant. It is produced as an intermediate of the Krebs cycle under aerobic conditions. Citric acid is mass-produced by continuous fermenter systems from cultures of the fungus Aspergillus niger. Carbohydrates are continuously introduced into the fermenter in order to maintain the citric acid production. Iron is excluded from the mixture in order to slow the further conversion of citric acid within the Krebs cycle. As the citric acid accumulates, it is extracted as part of the medium that is continuously withdrawn from the fermenter. Insulin is widely used to treat type 1 diabetes. It was previously extracted from the pancreas of animals such as cattle or pigs that were sent to slaughter. Insulin from slaughtered animals is not identical to human insulin and so is less effective than human insulin and is expensive to extract. In 1978, synthetic human insulin was developed by genetic modification of a bacterium. The gene for the human insulin was combined with a plasmid to act as a vector, so the gene could be inserted into the bacterium. The resulting genetically modified bacterium enabled the production of vast quantities of human insulin at relatively low cost. Insulin is manufactured by continuous culture. Bio Remediation is the use of microorganisms to clean the soil and underground water of polluted sites. The organisms convert the toxic pollutants to less harmful substances. The idea really started when a modified bacteria was made in 1971, which enabled the breakdown of crude oil. And it was proposed that this bacterium could also be used to treat oil spills. Solvents and pesticides can also be treated using bioremediation. Bioremediation involves stimulating the growth of suitable microbes that use the contaminants as a source of food. It requires the right conditions for the growth of the organisms, which are available water, a suitable temperature, and suitable pH. Where conditions are not suitable, they may be modified by the addition of suitable substances. In some cases, additional nutrients, such as molasses, may be needed to ensure the microorganisms can grow effectively. It may be necessary to pump in oxygen for aerobic bacteria. Where conditions cannot be suitable in situ, the soil may be dug up and moved to a treated ex situ site. Now, there are loads of advantages of bioremediation. It uses natural systems, it's less labour and equipment sort of intensive. Treatment can happen in situ. There are few waste products, and there's less sort of risk of exposure to clean-up personnel. However, bioremediation is only suitable for certain products, so heavy metals and lead cannot be treated.